to 10 o'clock, so I'm happy to get us started today. Welcome everyone here in person and everyone who is joining us online from around the world. It is my pleasure to welcome you to the 2024 Baruch S. Blumberg Prize Seminar. We have a very exciting speaker and topic this morning. My name is Shari Cohen. I'm the president of Hepatitis B Foundation, and I am very happy now to introduce our seminar speaker and our prize winner. So for those who don't know, the Blumberg Prize is the Hepatitis B Foundation's highest honor. It's named for Dr. Blumberg, who won the Nobel Prize for the discovery of the hepatitis B virus. The prize is given annually to individuals who have made significant contributions to hepatitis B, hepatitis delta, or liver cancer. This year's Blumberg Prize winner is Dr. Jake Liang. Dr. Liang is an NIH Distinguished Investigator. He is the Chief of the Liver Diseases Branch and Deputy Director of Translational Research at the NIDDK at the National Institutes of Health. He is a graduate of Harvard College and Harvard Medical School, completed his postgraduate training also at Harvard Medical School. Dr. Liang, as you know, is an internationally renowned thought leader and investigator in the field of viral hepatitis and liver disease. Dr. Liang's research program focuses on the virology, pathogenesis, vaccine, and therapeutic development for viral hepatitis and liver cancer, and he has published over 300 papers in the field. Dr. Liang has been associate editor for top journals, including Gastroenterology, Hepatology, and Gut, and he has been in various leadership roles, both at the NIH and the governing board and past president of AASLD. Dr. Liang's outstanding contributions have been recognized with numerous national awards, although this one's the best, including the AASLD Distinguished Mentor and Clinician Educator Award, the AGA Distinguished Achievement Award in Basic Science, and he has been elected to the American Society for Clinical Investigation, the Association of American Physicians. He's an AAS Fellow, also the American Academy of Microbiology and the National Academy of Science. So I feel like this award definitely rounds it all out for you. And we are very pleased now to introduce Dr. Liang. Just a quick note for housekeeping. We are going to, monitoring the Q&A online. So if you're on Zoom, please feel free to put a question or comment in the Q&A, and we will get to them at the end after the seminar. Welcome, Dr. Liang.
For that kind introduction, can you all hear me okay without using the microphone? All right, great. Well, thank you very much. Thank you for that kind introduction. I would definitely say this prize is indeed the pinnacle of my career. Thank you very much for the foundation. And it's always a great pleasure to visit this place. I enjoyed it so much every time I come here. So when somebody told me I'm going to get an award for expense trip here, of course I come up here. And it's great also to come to meet with friends and colleagues, and love to catch up with any of you after my talk. And let me just share a personal story with you. I was born in Taiwan, and as many of you know that at least back decades ago, Taiwan was one of the highest incidents of perinatal transmission of hepatitis B. Right? And I was born there, and fortunately, I escaped from that high prevalence of a perinatal transmission, but certainly some of my friends and cousins and uncles acquired it. So when I came here, when I immigrated here when I was a kid with my parents, I started to pursue medicine because that's what I love. And while I was at medical school, I had a needle stick. And back then, there was no catch-up vaccinations and all this stuff. That was in the early 80s, right? And then actually I got hepatitis B, but I recovered, fortunately. And this is something I really have not shared with too many people. But given this is a situation, so you can see that I have a really personal touch in this particular virus. And so since then, I've been trying to dedicate my career to beat this virus. I know, although I recover from the virus, I know the virus still in me somewhere. We all know. So I really want to be able to do something, and I know that I can't do it myself. I need to work with all of you to work together to beat this virus. I know get a cure for hepatitis B is the holy grail of the field, and certainly that's what Hepatitis B Foundations have been dedicated to do, to which I highly appreciate it. And so just share that piece, and I'll have more to say about the prize later at the gala event. So this is a photo for the ages. And I would say my career journey working on hepatitis B really came from my close association with Hepatitis B Foundation, who had sponsored this Princeton HBV workshop for many years. I don't remember when is the first one, but this is one of the earliest one. And you can see that this is why it's such a photo for the ages. We have two Nobel laureate and Dr. Blumberg, for which it's prize based on right there, and Dr. Alter, who's actually in the audience, and then many folks here who are the prize winners, such as Dr. London, Dr. Shinazis, and certainly we have our fearless leader of this workshop here in the front, the young whippersnapper here. I think much younger than here now, but just like all of us, and Brian McMahon and quite a few other people. So this has really sort of been part of my career journey with many of my colleagues for the last 30 years, and it's really been a great journey together. I hope I can continue to do that at least for a few more years, and to work with many of you to establish or to continue the friendship and collegiality we have had over the last 30 years. So you all know about hepatitis B, so I'm not going to tell you much about hepatitis B, and but just to say that this virus is actually prevalent throughout the animal kingdom, or related virus prevalent among the animal kingdom. You all know about the viruses infecting woodchuck, ground squirrel, and the duck. But some of the related viruses, especially the class of virus called negative and viruses, have been found in fish and amphibians. So this is certainly very much across the animal kingdom. So therefore, HPV-related virus really has a very close association with human history. There's been considerable controversy regarding HPV's evolution and timescale in associating with HPV. But I think from some of the earlier studies, actually studied back in 10 years ago, was actually the first case to show that we can isolate hepatitis B virus from a Korean mummy, which is about 350 years old. And then another study which came out shortly after in Nature really demonstrated the fact that virus is really quite old. And this is a study from a group of evolution biologists in Europe. They were able to identify or isolate the ancient hepatitis virus from the Bronze Age to the medieval periods. And they were able to isolate 12 cases from human remains from the Eastern Eurasia and Central Asia, in terms of be able to actually identify hepatitis virus in these human remains, and they were able to trace back to thousands and thousands of years. And as you know, that hepatitis B virus has multiple genotypes, and they've been spread across the world. And so it's probably these genotypes evolved with human migrations from Africa to the rest of the world, and resulting in many of these diverse genotypes. And I would say that hepatitis B virus is a virus antiquity, and is probably more than 400 million old based on certain kind of molecular clock estimation of the related viruses. The frequent cross-species transmission over time have resulted in widespread distribution of the viruses in the whole animal kingdom. And modern HPV evolved by a series of recombinations events with other hepatinoviruses in both primate and humans. So the entry of HPV into human is estimated to be about greater than 100,000 years, probably jumping from the primates as a Homo sapien evolved into a species. And so therefore, there is a considerable potential public health implication of cross-species transmissions. And but obviously the time scale is very long, right? Considering that. So we're probably not seeing much now, but we'll be surprised in another 100,000 years if the Homo sapiens still around, that you may see other new species of hepatitis B virus. But at this point, I would like to pay tribute or recognition to a close colleague and friend of ours, Steve Loccadini, who really had done pioneer work in this particular area and showcased how old this virus is. So make us realize that we're really just a small piece of the evolution of the virus, and human. So the HBV has a very compact and highly efficient genomes. I don't need to go through that, but just to share with you, probably every single nucleotide in this 3 kilobase genome encode more than one function. Just think about the overlapping nature of this particular virus. And the virus encodes 4 major classes of genes. The polymerase, which is important for the replication of the virus, the pre-S1, S2, and S are the envelope proteins, which coding the virus. And you have the core genes and core protein, which involve in the nucleic capsid formation of the virus. And then we have this protein called X, the accessory protein. So when you talk about an enigmatic protein of HBV, HBS come to mind, right? It's a small protein, accessory protein, has many functions. Sure, I mean, we all know that many viral protein has carried different pleiotropic functions and been likened to like a swift army knife. But in the case of hepatitis B X protein, this is really what's been in the literature. There are many functions associated with the X. And I would have to say that probably, yes, I understand the virus, this particular protein can have many functions, but I kind of doubt that it really had all these functions ascribed to this particular protein throughout the last 30 years of literature. So some of this is probably because of the system that we're using in terms of identify the function of this protein, that some of this may be artifact or maybe system specific, and may not be biological relevant. So regardless, we still really don't know enough about the X protein. What I can say for sure is that probably the most important function that X has been validated to have is interaction with the DDB1, which is a part of the ubiquitination, the colon, the ring ligase system that will facilitate the degradation of host restriction factors, the SMC5 and 6, which is important for regulation of HBV transcription. And this is probably predominantly what X does. Certainly X does many other things, but I just think that there's still a lot to work. But I'm not going to talk about X that much today. I'm going to talk about this other enigmatic protein, the E antigen, which, although may not have as many functions, has been assigned to HJVX, but it also been a lot of work that specifically identify various different functions of the E antigens. So I will tell you that my interest in this particular antigen or HBV began back more than 30 years ago when I was a resident. During which time I saw a 20-year-old patient who came to us with fulminant acute hepatitis B. And his risk factor is man having sex with man, and that's typically what the most common risk factor is. And there was really nothing much we could do other than supportive care. We didn't have liver transplant, we didn't have antiviral, so unfortunately he passed away. So that really had a real impact on me in terms of how serious this disease can be. And just about the same time, Edna Ben-Parath, who is a professor at University of Haifa, came to visit Jack Wong's lab, and I struck up a conversation with her. And she told me that, or just quickly define what fulminant hepatitis B is. It's a really acute, rapid onset jaundice, and typically with liver function tests, totally abnormal with ALT greater than 1,000, bilirubin greater than 20, and typically diagnosed by positive HBV serology for acute hepatitis B. And again, as I said, there wasn't much to do other than supportive care. There was no specific therapy or liver transplantation. So anyway, when I struck the conversation with Edna, she told me that she has identified a nosocomial outbreak of 5 patients with fulminant hepatitis B in Israel. So we began to say, why don't we study this? And she also identified a source of a chronic HBV as a carrier that spread into these 5 individuals. So back then, that's when we had PCR, right? So we could really easily amplify the viral genome from the limited amount of serum sample we have. So once we amplify this HBV DNA from the source and the 5 patients with fulminant hepatitis B, and we sequence them, and they have near identical sequences among the source and 5 patients. And what's most interesting about this, we identified two mutations in the pre-core region preventing the production of E antigens, which is kind of great interest to us because we thought, we have this viruses that cause fulminant hepatitis B. So does this suggest that E antigen may play a role in the etiology of fulminant hepatitis B? So we really began this journey to study this. At the same time, Masao Amada from Japan would also analyze a cluster of fulminant hepatitis B in Japan, and he derived at the same conclusion that many of these sequences indeed have mutation in the pre-core region that inactivate the production of E antigens. And we subsequently to these two papers, we studied further additional cases of fulminant hepatitis B from US in collaboration with many colleagues. We also identified a predominance of these viruses have the mutation, although I would say it's not exclusively. We certainly identify wild-type sequences in some of these cases, but there seems to be a predominance of this mutation in these fulminant hepatitis B cases. So let's go back to the core genes. There are really two proteins that produce one of these genes. Really, the fate of each is determined by which RNA encodes it. There are two classes of RNA that transcribe for the core genes. If you begin the transcription before the pre-core sequence, you basically give rise to the pre-core core protein, which is a secretory pathway. And the second RNA species that begin transcription in the pre-core sequence. So therefore, from there, you will give rise to the core genes, and that is the cytoplasmic pathway. So the cytoplasmic pathway produces a typical nucleic capsid core protein, the P21, which in combination with pregenomic RNA polymerase to form the capsids. And from within, that's where the virus replicates. And then in complex with surface antigen and the ER and Golgi produce infectious virus. So clearly the cytoplasmic pathway is essential for the HBV replication. The secretory pathway, because of this presence of this pre-C sequence, which contain a signal peptides that were directed to the ER and undergo cleavage by the signal peptidase to cleave out the N-terminal BC sequence, and that gets further transported to the Golgi, in which the furin will recognize certain sequence at the C terminals, which contain an arginine-rich sequence. So that will give rise to the secreted E antigen. That's a P17. But interestingly, there's another fate of this secretory pathway, which is really more the cytoplasmic pathway. This P20, the cleaved P25, could be retrograded transport into the spiking of the cytoplasm. And this is actually initially shown by Jim O and Ben Yan's group more than 30 years ago, that there's this species of cytoplasmic form of the pre-core protein, but they didn't know what the function was. And also this arginine-rich region in the C terminals contain a nuclear localization domain, so you can direct it to the nucleus. And the mutation we uncover reside in the pre-C region. So basically it's a stop codon mutation, therefore really totally abrogate the secretory pathway with the absence of any of these, either the P22 or P17. So what is the role of this P22? And back then, there was some indication suggests that the P22 may inhibit HBV replications. And this is some earlier study from Jack Wang's group. And recently, Jiangming Hu's groups have really done a very detailed analysis of this P22 form that suggests that perhaps what its role is to regulate the intracellular nuclear core protein of this particular pathway on the left that may be accelerate the degradation of the core protein and therefore perhaps be involved in the encapsulation and replication pathway. But again, the jury is still out. We really don't know whether this P22 really play an active role in HBV replication. So just quickly talk about E versus core, you can see that the significant overlap, right, of the sequence between the E antigen and core antigen. So why would they have such a different functions other than residing maybe in different part of either the cytoplasmic or the secreted pathway? But we do know that the E antigen has this P22 form, which can go back into the cytoplasm. So what perhaps just sort of a brief look at this, the sequence of the E versus core. Interesting that the E antigen in this uncleaved N terminals has a cysteine residue that can form intracellular disulfide bond with the cysteine residue at 61. Whereas if you look at the core protein, because it doesn't have this pre-peptides, this cysteine at 61 will form disulfide bond with another core molecule, so the intermolecular disulfide bond. So this gives us a clue that this may be the reason why E antigen and the core protein may function differently. And further study came from Paul Winfield's group when they resolved the structure of the E antigens. And this is the structure of the E antigen they have in a ribbon diagram. This is on the side view. They formed a dimer, just like core antigen does. And this is the top view. And when they compared the structure of E antigen with core antigen, because of this difference in the disulfide bonds, they actually look quite different, right? This is a core dimers that have the peak structure, and on the top view, very different. So this suggests that because of the subtle differences actually could indeed give us a different function between the two proteins, despite the fact they share a lot of amino acid sequences. So Thomas Balmer and Yoshi Kagawa, when they were with me, they conducted a serious study to study this particular fulminant hepatitis B strains, and that will probably, I'm not going to go into detail, but just in a nutshell, that they show that this mutant will replicate much more highly than the wild type virus. And interestingly, that the difference in the mutation, the enhanced viral replication, has nothing to do with this pre-core mutation. Because for us, we thought maybe that was the reason, right? But when they did a detailed study, mutate this back and forth, there wasn't any, not because of this pre-core E antigen negative mutation, but because of these two other core promoter mutation that seems to control the transcriptions and the viral of the viral protein in this pre-genomic area, contributing to the enhanced viral replication. So this was rather surprising to us, but regardless, this is what we had. And some of these core promoter mutations identified in these fulminant hepatitis B case have been described in other viral variants or HBV variants that have been associated with perhaps increased disease or HCC. But again, the literature that's sort of unclear whether this core mutation really play a role in the pathogenesis of hepatitis B in humans. So quickly summarize this whole field that if we look at the clinical correlates of the E antigens in terms of HBV infection, and some of the animal models that have shown us infection with HBV pre-core mutant, this has had a mutation in the pre-core peptides, often associated with more symptomatic acute hepatitis, such as fulminant hepatitis, as I have alluded to, but less chronicity. And infants born to E antigen negative mothers are likely to develop chronic infection. So it suggests that E antigens play some role in terms of disease severity. Study in the animal model using either the woodchuck or the chimpanzee, basically mostly the work from Paul Cody and John Jaron, as well as Bob Tennant. When they introduce mutation in woodchuck, again, they also saw that there was much less tendency for chronicity in woodchuck. The same thing in chimpanzee when Bob himself to conduct the same study using this E antigen or pre-core mutants virus to put in the chimpanzee, it's less likely to develop chronicity. So this really suggests that E antigen would play a role in terms of chronic chronicity of HBV. So we were left with this E antigen enigmas. So what are some of the issues, right? Because we have both secreted and cytoplasmic clone E antigen. So does this function related to the extrinsic pathway, the secreted E antigens, versus intrinsic, which is P22, the cytoplasmic nuclear form? Or is this the E antigen effect generalized versus HBV specific? Is it, if it has an immune tolerance effect, does it affect other aspects of the immune system, or whether it was HBV specific? So these are really the key questions we have not been able to answer. So perhaps the whole idea about E antigen as immune tolerant was validated and revealed using the transgenic mouse model. That Dave Milich, back in the 1990s, conducted some seminal experiments, was able to show using transgenic mice expressing E antigen, showing that the E antigen as a small soluble protein can pass through the placenta and induce immune tolerance toward HBV. So this is really a direct experimental evidence suggests that E antigen is immune tolerant. And subsequently, David, working with Frank Cesar's group using various different HBV transgenic mouse model, can further validate that the E antigen's role can actually polarize the T cell response to its related core antigens. And that's also, again, explain why E antigen may play a role in terms of the induction of a chronicity. So I'm just going to kind of quickly go through some of the current hypotheses about the function of HBV and show you some of the evidence. And Anna Krampus group had done a really masterful review articles summarizing all the proposed function of E antigen in last year's publication. So looking at this particular, some of the study she had referenced here, that the E antigen could interact with peripheral blood lymphocytes to increase the inductions of myeloid-derived suppressor cells, which can have a suppressive effect on the immune system. So this is one of the theories that was shown from the Jingming Hu's group in Fudan. And Jim O's group, on the other hand, again using the mouse model, they were able to show that the E antigen actually can polarize the macrophages from the M1 to M2. Because many of you know that the M2 phenotype macrophage tend to have a more immune suppressive roles, and therefore this may explain why the E antigen has its immune suppressive functions. And furthermore, a couple other groups also show that E antigen can somehow interact with the TL toll-like receptor family by causing some kind of immune suppressive effect. In terms of the cis effect, the intracellular form of the E antigen P22, high-tau growth groups were able to show very nicely that this precursor P22 can interact with the cellular protein K-alpha-1, which acts to block the nuclear translocation of the p-STAT1, therefore to reduce the JAK-STAT pathways, which is mediated interferon response. And in parallel, another group suggested that the E antigen actually can upregulate SOX protein, which is one of the protein will suppress this JAK-STAT signaling pathway to block the interferon effect. And there's also various different study interesting suggests that E antigen can modulate the microRNA pathway that can have much profound effect in terms of upregulation of kappa B, the MAP kinase, et cetera, that are causing so-called some of the downstream effect. There's also a couple other papers demonstrate that E antigen can actually interfere with the proliferative signal pathway of the cells, such as interfering with the RB protein pathway, as well as the Wnt pathway. This is some of the mechanism that was proposed in these studies. And lastly, that E antigen has also been implicated in regulation of apoptosis. And using through the external pathway, the secreted E antigen has been implicated to associate with the Fas12 ligand pathway in order to modulate apoptosis. But the internal intrinsic pathway study also suggests that P22 can interfere with the P53 pathway, which again kind of makes sense in terms of proliferation. And lastly, that Anna's groups was able to identify another mutation in the pre-core region of the E antigen that affect the cleavage of a signal peptidase is a valine to phenylalanine mutation. And what they observed that in the cells transfected with this particular construct, there was a reduction, increase in the unfolded protein response in terms of ER stress that can lead to apoptosis. So when you have this mutation, there's reduced unfolded protein response, therefore increased survival HBV. This is actually reminiscent of this other study that we conducted quite a few years ago, showing that the hepatitis B surface antigens can activate unfolded protein response in forming what we call the ground glass appearance, which for many of you who are clinicians realize that this is a histological hallmark of chronic HBV infection, that these ground glass cells are packed with surface antigen. They tend to die from the overexpression of the surface antigen. So anyway, in this study that we took advantage of the adenovirus expression vector generated by Blumberg Prize group, and she has all these adenovirus expressing either just GFP, the HBV, the surface antigen, E antigen. So you can see that this is a very helpful set of tools to study the effect of individual viral protein. So using the adenoviruses, she had generated YALS, was able to actually, you can see that if you can generate very high level of infections of cells when you're using the adenovirus, evidenced by pretty much uniform positive GFP signal in this cell culture system. So we can do the studies and we can look at HBV marker and look at unfolded protein response related to the ER stress. And YALS were able to conduct this experiment by infecting cells with the different constructs. And she looked at 4 ER stress markers. So, okay, the BIP, ATF4, CHOP, and GAT34. These are pretty typical ER stress marker people study. So she has a positive control when she treated the cells with a protein translocation inhibitor cocktail, which has been shown to significantly induce ER stress. So that's a positive control. So among all the construct that she infected the cells with, only the surface antigen expressing adenovirus highly induced ER stress markers all across the board. And she's further studied looking at the surface antigen expression. Indeed, the surface antigen expression construct produced high level of the surface antigens intracellularly. And there is one construct she used that produced a full-length HBV, which does produce low level of surface antigen, but certainly not to the level of the adenovirus that directly increase the expression of surface antigen. And then in this particular experiment, indeed, the BIP ER markers significantly induced in the surface antigen overexpression cells. And then this is just a supernatant of surface antigen. Indeed, the anti-S produced high level of surface antigen in the surface, in the mediate, and again, presence of the big BIP stress markers. So the question we have is that, does E antigen suppress ER stress induced by surface antigens? And we could kind of do the mix and match experiment, right? Kind of infect cell with 2 different viruses to see whether that's the case. And that's exactly what Liang did. So she would mix the adenovirus in different combinations. And so certainly the critical experiment is to infect the cells with both E antigen and surface antigen to see what happened. And this is just checking out the testing the HBV markers. Indeed, the adeno-8S and can produce high level of surface antigen, E antigen, adeno-E can produce E antigens. There was maybe somewhat suppression of the E antigen production when you co-infect the cells with surface and E antigen containing adenovirus. So when she looked at the ER stress markers, and only 2 of the markers were positive, right? Because she was using lower MOI, so that's why maybe she didn't see the ER stress induction on the other marker. But regardless, if you look at this particular 2 ER stress markers, CHOP and GAT34, again, with surface antigen expression, there's a high level induction of these 2 markers, and none with the E antigen expressing cell only. But when she co-infect E antigen with surface antigen, there was a significant reduction of of the ER stress marker, suggesting that maybe what E antigen is doing is blocking the unfolded protein response induced by surface antigen. So that's certainly a very attractive hypothesis and consistent with some of the previous study I quoted. So, but we're still skeptical about this particular question. So, first of all, ER stress was only induced by very high level expression of surface antigens. And does natural HPV infection actually reach that level of surface antigen expressions? Probably not in the natural infection, but possibly in some situation when you have cells that express high level of surface antigen, as in those ground glass hepatocytes that I described to you, but certainly not true in terms of the natural HPV infections. And finally, it's a suppression of ER stress by E antigen general in terms of the overall ER stress response, or specific to HPV, and how? What is the mechanism? So that's really the remaining question we have had. So at the same time, we've been sort of a little frustrated using some of these surrogate expression systems, right? Adenovirus, transfections, all this stuff. So we really thought that we need to go back to much more biological relevant infection systems. And as you all know, that we do have the virus that we can isolate from cell lines such as 2215, 8038, which produce infectious virus that can, in fact, that can be shown to be used to infect naive cells, to do various different studies. That's what we all do. But unfortunately, that's only one genotype, right? That's the genotype D. That's what everybody uses. And we really don't have any cell line that express other genotype viruses. As I mentioned to you, there are many, many different viral genotypes. So we can't really just study one genotype virus and trying to reach sweeping conclusion across the HPV, because there's some good indications suggesting that genotypic difference probably exists in terms of the disease expression, treatment response, and other clinical manifestation of the HPV. So we thought we're just going to go ahead and try to produce various cell lines producing different HPV genotype. And this is what Ming and ZZ did in collaboration with Peter Revels from University of Melbourne, who has collected a large number of isolates of all the different HPV genotypes. And also with Professor Chayama Hiroshima, who has this wonderful facility of humanized liver chimeric mouse model, which we can actually infect mice with very different HPV and demonstrate infection, which I'll show you later. So anyway, this paper really described more of a methodological paper talking about the generation of the different cell clones. And we have generated this, and now they're widely available. They can be available for ATCC. If anybody want to use it, just you can request them. And these are the features and described in this particular paper. So just a sort of brief summary from this paper that was published in the Journal of Hepatology a few years ago, that the virus generated from these various genotype containing cells can be stably passaged in both in vitro and in vivo model. We have shown that virus generated here can infect the humanized chimeric mouse model. And then these HPV genotypes, interesting is that do exhibit different infectivity, antigen expression, replication, and treatment response. So this really suggests that phenotype is not just because of difference in terms of sequence, but there are differences in terms of clinical phenotypes. And then these models we feel are a valuable tool to study HPV biology and also antiviral development, right? Because if you want to develop antiviral, it needs to be cross-active against all different genotypes. And all the tools we have right now is just against genotype D. So I think this is would be hopefully be an important tool for all of you to use in the future. And what's also interesting coming from that study is that when we compare the virus generating cell culture versus the virus passage in this humanized chimeric mouse model, we call it HBV-NP, okay, it's a mouse passage, and the HBV-CC is cell culture generated. So we compare the characteristic of the virus from both of these sources, and using density gradient analysis. This is a density profile of the HBV cell culture generated virus. And the black line represents HBV DNA, we measured the different marker at different fraction of this gradient. The HBV DNA is black, surface antigen is red, and core antigen is blue. And the Dane particle, which is infection particle, typically reside in fraction 10. All right, you can see that it's there. That's good. But surprisingly, that there's really actually quite a bit of what we call a naked nucleic acid that's produced in the cell line, which is, again, is not new. People have described that contain pretty good level of HBV DNA. So these are kind of a sort of just a feature of a cell culture DNA virus. You do produce these sort of nucleic acid negative proteins that is not completely virions. But when we analyze the HBV pathogen in the mouse model, and there was a very clear defined peak of Dane particle that contain HBV DNA, surface antigen, and core antigen without much aberrant fraction of core particle. And what's also interesting is that when we infect naive cells with either HBV-CC and HBV-NP with the same MOI, the HBV-CC was much more infectious when we standardize based on HBV DNA, and which is you can expect because HBV-NP contain a really mostly infectious virus, which HBV-CC do not. And HBV-NP actually very consistent with what we see in infected humans, right? When you analyze the same, if you do the same analysis in HBV infected humans, you see predominantly this Dane particle fraction that contain predominantly HBV DNA. So we thought this is really a more attractive feature to use virus that passage in this humanized chimeric mouse model to study, to do a subsequent study. So in order to study the pre-core mutant further, so ZZ and Ming introduced the start codon in three genotypes that we have: the genotype E, genotype B2, and genotype C1. And again, using the same protocol that we described in our previous study, to be able to generate cell line producing high level of these viruses. So when we decided, they passaged these cell culture generative viruses in the human liver chimeric mouse model. And this is just an example of 4 or 5 mice here infected with the genotype C1 and the C1 pre-core mutants. All right, so because we want to be able to completely compare both of them, they are genetically identical other than this pre-core mutation. That's the only way we're going to sort out what the importance of this pre-core mutation is. So this is the time course of the infections of genotype C1 and pre-core, and this is a time course of genotype D and its corresponding pre-core mutations. And you can see that the viral titer marker elevate gradually and then plateau at around 6 to 8 weeks or so. And it's pretty typical what we see when we infect these chimeric human mouse model with HBV. And also what's validating is that indeed in the pre-core infected mice, there wasn't much E antigens as you would expect, right? Because the E antigen should have been inactivated. And so when we combine all the data together, just see whether there's any difference in terms of the infection course between the core and pre-core mutants. As shown here, it was, pretty much no difference between the pre-core core mutations in terms of HBV DNA production between the genotype, among the genotypes. And then we look at the surface antigen a little more carefully. Interesting enough is that the pre-core mutant seems to produce a little higher amount of surface antigens. Seen here in the genotype C, although not statistically significant, but clearly significant in the genotype D case. The surface antigen level is higher in the pre-core infected virus, which is interesting. We don't know what the reason for that is, but this is clearly reproducible. So there's been a lot of discussion about whether these pre-core mutant, whether they may be more or less sensitive to antiviral therapy, because there's been a lot of study in the clinical arena suggesting that the E antigen negative virus may be more or less sensitive to interferon or other. So anyway, we thought this is a perfect model system to look at the response to interferon, because there's no immune system in these mice, right? We can see whether there's any intrinsic difference in terms of the susceptibility to the interferon in this model. So Ming conducted this experiment and showed, see, so this is just, so we treated these mice with 4 weeks of pegylated interferon alpha 2. And you can see that there was, during the 4 weeks, there were significant reductions of HBV DNA level in most of these mice, in all these mice actually, and for genotype C1. And similarly for genotype D3, there was a reduction of HBV DNA, although the magnitude of reduction is somewhat lower in the genotype D3. So when Ming put all the data together, trying to look at the change in HBV marker using area under the curve analysis of the 4-week treatment, and shown here on the left, on the right, that indeed that the HBV DNA. So if you look at the HBV DNA of all these mice, in terms, there wasn't really any difference between the wild type and the pre-core mutant, either in genotype C1 or genotype D3, and we didn't see really actually much effect on e antigen versus surface antigen. Well, e antigen obviously doesn't produce e antigen, but we didn't see much effect on the surface antigens. But regardless, if you look at just the antiviral effect on the HBV DNA, there wasn't any difference in terms of susceptibility to interferon between the pre-core mutant or the wild type. So we can also do the same experiment in the cell culture model, right? Because we can take the virus passage in the mouse and then infect the PXB hepatocyte. PXB hepatocyte, these are cells commercially available. These are the primary human hepatocyte passage in these chimeric mouse model. You can isolate them. You can do them in cell culture study, which really provide a consistent source of human hepatocytes, and they pretty much mimic everything that primary human hepatocytes do. So we could do the experiment in cell culture as well. So Ming infected these mice with HBV mouse passage virus, and then treat them with either interferon alpha or entecavir. Entecavir is nucleoside analog. And then she will harvest time points at different time of the culture and look at the HBV markers. And this is a 3 genotype B2, C1, and D3. You can see that they all get infected pretty nicely with these mouse-generated virus with gradual increase of HBV DNA here of the 3 genotypes, and they also quite susceptible to both interferon alpha and entecavir in terms of HBV DNA level, and there wasn't really any difference, right? That's a key. We want to see if there's any difference in the cell culture model. Again, there wasn't any difference. So it suggests that whatever people claim that there may be the difference, it's not true, at least in the cell culture model. There was no difference in terms of susceptibility of the pre-core versus wild type. Suggests that e antigen probably doesn't play a role much in the interferon response. So just sort of group all these data together, we look at HBV DNA and HBV RNA and surface antigen. These are the 3 markers we typically follow. So if you look across the board and looking at genotype B2, C1, and D3, and then compare that with pre-core mutant, there wasn't really any difference in terms of the reduction of viral markers across all the genotype. We compare the wild type with the mutants. What's sort of interesting is that when we look at the difference between genotype D3 and D3 pre-C, as I mentioned to you before, we saw a difference in the surface antigen level in our in vivo model, right? And this is also reproduced in the vitro model. Indeed, the genotype D3, at least D3 with a pre-core mutant, it produced higher level of surface antigen cell culture. We don't know what the reason is, but that's sort of a reproducible in both in vitro and vivo model. So finally, we have the system now. We actually can address some of the question of what is the ER stress, right? Does it really happen in a natural infections? And we have both wild type and mutant. We can directly demonstrate whether there's any difference in terms of ER stress or whether the virus does indeed induce ER stress at all. So Ming conducted this experiment, infected PXB hepatocyte with HBV mouse passage virus in the presence of no absence of Macludac B, which is surface of control. We want to block the viral infection. Also, she would infect a different set of, also treat a different set of infected cells with entecavir, just as because entecavir really mostly affect the HBV replication, doesn't affect the production of surface and E antigen, right? So that can distinguish what's causing the effect if there is indeed an ER stress. So this is just, again, the time course of genotype D3 compared to the wild type and pre-core. And as you can see that there's certainly with the Macludac, President Macludac, there's absolutely, no infection at all and across all the markers. And entecavir just blocks some of the surface antigen, but certainly not as much with the, this is probably because entecavir in this particular system can prevent the spread of the virus. It doesn't necessarily prevent the production of surface antigen, but if you prevent the spread, you're going to see downstream effect of the production of surface antigen. But nevertheless, that seems these are what we expected. And if you look at the effect on HBV DNA, clearly it's entecavir is very effective in terms of blocking HBV replication. So when we look at HBV DNA, right? So now this system works. So how about the ER stress markers? So we can easily do the experiment, and that's exactly what Ming did. She looked at the ER stress marker at different time points, day 4, day 7, day 10, day 14. And these are all the different markers and different conditions. And I really don't need to take you through it. You don't see any difference, right? We don't see any induction in ER stress in this naturally infected cell culture model. And also, there was no difference between the wild type and pre-core mutant. So this is really refute our hypothesis that the E antigen probably play a role in terms of the reduction of ER stress induced by surface antigen. So this is where we are right now. And so how do we pursue further? I mean, there must have some effect E antigen, otherwise we won't see all these clinical manifestation, right? So we decided to take on more of a holistic and unbiased approach. So we have these viruses, we can efficiently infect the cell culture system. So we do that. The key is that when we do that, we can isolate RNA from these infected cells and perform RNA-seq to see whether there's any difference in gene expression and performing bioinformatics and do functional studies. So just show you some preliminary data that when we plot the RNA-seq data comparing the D3, the wild type versus the pre-core mutants, and we clearly saw enrichment of certain genes that's upregulated in the D3, not in the P3, and vice versa also. So we have a sort of a cluster of genes that are differentially regulated between wild type virus infected and pre-core mutant infected virus. So this may give us a clue in terms of what is the downstream mechanism of E antigen. And then just running some bioinformatics using the GSEA, and there are various different pathways that enrich. And not to take you through it, but just to highlight some of the one right there: TNF alpha and kappa B, apoptosis, P53, KRAS. These all kind of came up in my previous review of all the literature, right, about some of the effect of E antigen people have proposed to affect the cell physiology. So this certainly gives us a sort of a rich trove of information that we're going to try to follow up and see whether we can indeed identify the precise mechanism. What does the E antigens does to the cell to activate some of these pathways? So I'm really done with my sort of showing you some of the data, and this is just some of the unpublished data. And certainly, as you can see, we have a lot more work to do, right? We still don't know exactly what it is doing. So I want to go back in time again to share with you this particular study from Taiwan. As many of you know, that Taiwan has play an outsized role in terms of studying the natural history, epidemiology, pathogenous hepatitis infections. And from the earlier study from Palmer Beasley and his Taiwan colleagues, who was able to definitively demonstrate the linkage of chronic HBV infection with hepatocellular carcinoma. And that's probably the first direct evidence of a human virus can cause cancer. Right? So obviously there are others now. So this is really quite impressive data, and And I really want to pay homage to Dr. Denchen Chen, who was a previous awardee of the Blumberg Prize. And unfortunately, he passed away a few years ago. He's been really a great mentor and colleague and friends, and I sorely miss him. And he actually has been really instrumental in changing the direction of HPV infections in Taiwan. As I mentioned, Taiwan has a very high prevalence of HPV infection. So he set up this community screening project and looking at the different factors. And he was also instrumental in implementing the vaccination, universal vaccination in Taiwan, which had led to significant reduction of transmission of HPV in Taiwan. So this is really a paramount public health importance. And so he certainly should be acknowledged for it. But anyway, in this study, this was, again, 20 years ago, that suggests that E antigens, which we all be interested in, really play a role in the hepatocellular carcinoma. We didn't know what that meant, and maybe this is a tumor marker, maybe it's an oncogene, whatever. But probably what it is just a surrogate marker for HPV replication level, right? And so the higher surface antigen, you will have higher risk of HCC. And I wrote an editorial back then about E antigens. I called the dangerous endgame of hepatitis. I wrote it together with Mark Dining, who was here. And I didn't know what that means, but I just thought that this is where the link is. But it's probably not a dangerous endgame, but certainly play a role in sort of the consequence of chronic HPV infection. So I'm really coming to a closure of my talk, and I just want to ask you, what is this? This is it's a time-lapse photography of the last eclipse back in 1945. All right, and I think you're all aware of it, that next Monday is the solar eclipse. I hope you got your glasses and you can go look at it. And but back then, in the time of Antiquity, it's considered as an enigma, right? I mean, all the ancient people, they see this. The sun gets swallowed by something, and they have no idea what that is. So there's been a lot of myth and stories. Actually, it's interesting if you ever want to find out, Google it and see what the ancient people think about the eclipse. And I think it's really a wealth of information, sort of interesting. So back then it was considered an enigma, right? A real enigma. How does the sun get swallowed by something that they have no idea what that is? And I think probably I would say for E antigen, at least in the context of my presentation, was an enigma back then, but I think we started chipping that away. We haven't really quite gotten to the point that we know what causes solar eclipse, but maybe another 30 years, hopefully not, that we will know exactly what E antigen does in terms of the HPV infection and pathogenesis. So I just want to close by recognize a group of people I've been working with. I've really been blessed with working on a large number of trainees and fellows and faculty member, research nurses and clinicians in the liver diseases branch. And it's really just been a wonderful ride and journey. And last night we were talking to some folks about why did I stay at NIH so long. This is the reason, because you have to enjoy what you do. And I love what I do, and certainly have the opportunity to work colleagues such as Harvey and Mark and others, and just been really a wonderful journey. And I hope I can keep doing that for a little bit longer, but I don't know how much longer. But anyway, I want to stop and maybe take some questions.
I want to point out, yes, thank you very much for the great earthquake type talk. I don't know if you know where you were speaking. You noticed the projector was shaking, the room was shaking.
Well, actually, I forgot. I do want to pay sympathy to what's going on in Taiwan, right? You'll know about the earthquake, right? A couple of days ago, and I reached out to some of the colleagues I know well there. They said they're okay, but obviously the island is devastated in many ways. So I do want to kind of maybe give a minute, kind of think, because
We did So for those who noticed that the room was shaking, there was a 4.8 Richter scale earthquake here. During Jake's talk was earth shattering.
You have to find sympathy.
But I do, just in case that I've been checking in with Lou, the building's okay. Hopefully everybody's been able to check in with family and friends and are doing okay. And yes, we definitely we've also checked in with a number of colleagues and board members and friends who are all in Taiwan and are all thankfully doing okay. Happy to open it up to
Well,
questions.
I do have a question though.
Yes.
So, two questions, one little one, one bigger one. The little one was a technical one, where they were looking at, e-antigen suppression of the unfolded protein response. And was it ever determined whether or not there was a trans effect on there was a control in there, I guess, the DTT or something that induces, unfolded protein
Yeah.
response?
Yeah, we haven't done that to see whether E antigen will suppress the ER stress induced by the protein translocation inhibitors cocktail, which is standard use for induction of ER stress. Yeah, to see whether it suppresses this other or whether it just surface antigen. Yeah, I mean, that's a good question,
There's
but
could be some artifacts in it.
yeah, it probably is, because certainly, based on our infection data, we didn't see any induction of ER stress, so.
No, so, and then, so a couple more questions. In the ground glass hepatocytes, is that, I guess you implied the level of S antigen is very, very high, and the level of E antigen is very, very low. And then I'll ask my second question. What's known about the metabolism of E antigen? I mean, in the circulation, as it's picked up, when it doesn't, when it's no longer in the circulation, E antigen. So first one was a simple, straightforward one about ground glass hepatocytes. I said S antigen is very high, E antigen very low.
Yeah, well, nobody ever looked at that, whether the expression of E antigen. I guess it's hard to detect E antigens, right? Because it's mostly secreted unless you have a very good specific antibody that will pick up this pre-core form, right? Because that's really what it referred to, right? And there are antibodies like that. And I don't think anybody look at clinical specimen looking at these ground glass hepatocyte, whether they're indeed much low expression or presence of these pre-core related proteins. That would be a good question. Maybe that's the reason why these ground glass hepatocyte are producing huge amounts of surface antigen causing ER stress and cell death.
And the metabolism, how's E antigen cleared?
Yeah, I think it's probably just like any protein gets picked up by RES systems and degraded and stuff like that. So it's actually I can't even tell you whether it's a half-life E antigen. So I don't think anybody done that. And also another thing you can be aware that even though for patients with chronic infected HBV, they probably still produce low level of non-neutralizing E antigen antibodies. So there's probably some small amount of antibody complex between the anti-E and E, although there's probably much higher E antigens in chronic infected individual than antibody, so you don't see it. But that may contribute to some of the turnover of E antigen circulations. But yeah, so anyway.
Thank you.
Based on your adeno S expression, it looks like it's small surface only, right?
Yes, it's only expressed small surface.
Yeah, large, medium, and small together, and whether they still induce ER stress.
Yeah, I mean, we haven't done that, but other people have. Pre-S actually has been linked to when you overexpress it, induce ER stress retention on the surface, and because the pre-S doesn't really get secreted by itself, right? It gets retained. Actually, Frank Cesaris did a lot of work related to transgenic mouse models expressing pre-S, and they get these these cells that express high levels of pre-S in the hepatocyte causing cell death and others. So yeah, I mean, I certainly think that pre-S is also a culprit as well. And we haven't really done that just because, again, my bias that when you're using this high expression system, you're looking at artifacts. It may not be biological relevant. So that's why we are really moving toward more sort of the natural, more biological relevant infection system. And that can give us probably really what's happening with the infectious virus.
So,
Go ahead, sorry.
so very nice. You nicely reject all the possible effects of the E antigen intracellular.
Well, I'm not rejecting it.
Yeah,
I mean,
basically previously proposed this effects.
It's from the P22, the intracellular.
Yeah,
Again, it's just still debate in the field and whether it's the extrinsic, the secreted E antigen affecting all the,
Yes,
right?
that's what's where I'm going.
Yeah.
Basically, for a long time, I actually suspicious about, the E antigen really does that really have effects inside the hepatocytes? It's, their real function is after secretion. It's actually, the secreted E antigen, they do have all those effects, right? And your in vivo study, I think that was pretty consistent with what Jesse Summers have did, 20 years ago with DHB.
Yeah.
And what he did is actually use E- virus and the white type virus mixed, and then do this in vivo competitions.
Yeah.
And with basically no one win. They're all
There's no, yeah. So I think the effect is subtle. I'm not saying it's very, very clear, but it must have some effect, right? Because
Yes.
from all these studies. So
Well,
the
that-
question is, wait, what?
Yeah, I think, evolutionally, this precore protein as, that's also, I also consider as, accessory protein, right? Just like X, we take several decades to figure out its function. And for this accessory protein, it starts from what brains, right?
I'm sorry?
The HBV-like virus in
Yeah.
what brains? Like
Yeah.
and mammalian, they start
Yeah,
have this
yeah.
precore protein.
Yeah.
They even appears earlier than X protein, right? So they must have some some function.
Yeah, otherwise evolutionary will
Yeah,
kind
so
of
I
activate, right? So,
And also another very interesting thing is that if you look at HPV infection, every chronic carrier going to experience this Y type to the E- virus conversion.
the
someone-
vice versa too, right?
Yeah.
Yeah.
Somewhat early, somewhat late.
Yeah, no, I agree. There's a lot we don't understand what actually happens. Sorry, I think she had the question back there.
Well, I mean, it's really a great talk and then a lot about E-antigen, okay? But I think it's very exciting to see how, so many years ago, E got linked to hepatocellular carcinoma. And now you're showing, I believe, if I got it correctly, with the E-antigen, the one with mutant, one without mutant, they grow in about same level. But the one with the one without mutant that activate the P53, is that all the opposite?
Well, that's a hypothesis from earlier study.
Yeah.
Specifically looking at the
Uh-huh.
P53 pathway.
Okay.
In the antigens, and there are some papers indicating that. But also, what we saw is the pathway that seems to be affected.
Yeah, the pathway.
So we don't know whether it's really P53 or whatever, right? Because
It's
usually when you do these bioinformatics, they do a lot of sweeping type of
Uh-huh.
pathway. So we have to look at more detail, look at particular genes that's differentially regulated and see how they are connected to the actual mechanism of the E antigens.
But in that RNA-seq study, the viral, in terms of the viral replication, they are.
It's the same. I mean, I showed the time course of HPV.
Yeah, but the
Like the
pathway-
biomarker, that's the same, and we have done staining, they all look this, the same between the mutant and the wild type. So it's not because of difference in viral replication. And it's, we think it's because of the E antigen. Obviously, there's a lot more work to do, right? Once we find whatever the pathway or genes that differentially regulate it. Although the good thing is that we have other genotype, right, with the core mutants, right? That's the reason we did this, because we don't really want to just focus on one genotype, because it's true, it should be across all the different genotypes, right? So that's why we have these two other genotypes that we make the same mutants, and we are studying the same way. And if this is indeed real, we should see across all genotype, right? If the whatever pathway is affected by E antigen, we should see that in all of it. And that's really our hypothesis, right? If we can't show that, I don't know, maybe genotype specific, I guess could be, it could be
Yeah,
genotype
it's
specific,
because
could be.
the-
And we did see a difference in surface
Somehow
antigen.
also linked
Yeah, we
to
did see
contact.
a difference in surface antigen
Uh-huh.
expression, only in genotype D, right?
Okay.
Not much in other genotypes.
Uh-huh.
So there may be some surface, some genotype specific effect but I think the general effect should be across all the different genotypes. Right, that's my bias, so.
Yeah.
That's, yeah.
No, I think it's interesting because we also try to look at the integration with this E antigen positive, negative, and for some reason, when we look at HCC recurrent integration, out of that 30-30, we only see the E antigen positive survivor. It's just
Yeah, that maybe have to do with
the
the
stuff-
viral replication
yeah.
level, right? I mean, that's the integration, so, I'm sorry, Mark?
So something that's always sort of puzzled me, Jake, is for acute infection, you need to have E antigen. Is that true? And why is that?
For acute infection, no.
So
I mean, that's what we show with these, outbreak of,
But I think you need to still have E present.
no.
To,
You can get productive infections with E antigen negative mutants, been shown in very different animal models and woodchuck, you say duck and chimps with Baruch S down, that's why I reference those. So for acute infection, productive infection, you don't need E antigens. But for chronic infection, you need E antigen to shift the immune response to immune tolerances, therefore more likely to develop chronic infections.
but these E negative mutants can cause chronic infection. So presumably they're still producing some small amount of E that's required.
Again, it's not totally clear. I mean, what's clear is that at least in the animal models that people have done, it's really almost mostly acute infections. Given the limited number of animals, I mean, Baruch S has done that, but how many chimps can you infect, right? So 2 chimps, so can you say they all acute versus chronic, right? So this is just an indication that suggests that what the E antigen is shifting the immune response to HBV to allow more chronic infection, but it's not absolute, right? We know that 90% of people, adult people, recover from acute hepatitis B, right? So it's not absolute. It's just that there's a shift of balance and with more of a chronicity. Yes, Harvey?
From a simplistic clinical standpoint, we've always assumed that E antigen positive people are more infectious in maternal fetal transmission. And so is that just a correlation with HBV DNA, or is there something else that it's doing to enhance transmission?
I think so. I think it's more to do with replication, probably predominantly due to replication, higher HBV DNA level in these mothers with E antigen positive. But the question is really the E antigens may play a role in terms of the placenta crossage, which has been shown. I mean, the E antigen is small enough that can pass the placenta to the fetal circulation. So I think that's kind of what David Milligan originally proposed makes sense to me, that the E antigen does cross the placenta, polarize infant to core-related antigen, right? And then so when the infant gets exposed to the virus, probably usually during delivery, right, process, that the infants is somewhat tolerized to the core response, so therefore they may be more likely to develop chronic infection. I mean, that's a hypothesis. And it kind of makes sense because that's what, again, Jim Mo's group has sort of shown in his recent study that it sort of a shift M1 to M2, causing more immune tolerance effect on the fetus. Right? So I think that's kind of the hypothesis. Right.
Jake, thank you. Hey, it's great talk and lots of great questions, and so it was obviously so good, made the earth move. But moving on, the HB actually talked about it earlier. I'm definitely interested in immunology, and I would like to explore your thoughts about what if we're able to induce highly specific, high active antibodies to X in the form of a therapeutic vaccine, coupled with other agents. That's what your thoughts on looking forward on that might be.
Randy, you asked a really kind of fascinating question. Really, can X? What do we know about X?
Yeah.
And if we do more about it, can we leverage that knowledge to turn antiviral? right? I mean, it sounds like you're talking more about sort of the immunotherapy.
Yeah,
part of the
therapeutic
X.
vaccine, yeah.
I mean, unfortunately, X protein actually is not generating abundance infected hepatocytes. So actually, most of us had a difficult time identifying X protein in a lot of the system we use. So I think whether that could potentially be a target for either immunotherapy, either CAR T cells or the T cells, TCR receptor stuff. It's possible. I mean, I think it is, but most of the people working on that field are really targeting something much more common like surface antigen, kind of using that. So I just don't know. I mean, I don't think anybody. It's the X fields. I mean, it's Utah can agree to. It's just really full of controversy. I would say probably 90% of the thing we read in published probably on artifacts. So, I mean, but I do think with some of these more biological relevant systems that we start using, we can generate very useful information. But for example, what I alluded to about the DDD1, the ring column complex, that X target, I think that's true real. I mean, Jude, do you agree with me? I think that's real. So, but I'm not sure about 90%.
People have started looking for disrupt that interaction, which looks like very difficult to.
You mean for anti-X? Yeah. Thanks, Randy. Thank you.
Two questions from online, talking about HBX and HBe antigen Enigmas. The duck and all other avian HBV do not encode X, but all have E antigen. Was wondering why no VCP or PC mutation found in DHBV. What are your thoughts about this?
Oh, no pre-core mutations in the DHBV. Well, I mean, maybe Jude talk answer that better than I do, but I think the duck system is just very different from the mammalian system, avian system. They tend to have a different disease manifestation. They have good high replication. They don't see the kind of disease we see in for instance, human, even the woodchuck and the human virus is different. So I think maybe just unique to the avian papillomaviruses and maybe the way the virus evolved in the avian species, it doesn't need X protein or whether this pre-core play a role. I mean, again, the transmission is different, right? I mean, the avians, you're not talking about maternal fetal transmission, right? It's an egg, and so it's very different from what we face in terms of the physiology of mammalian system in terms of the maternal fetal transmission, right? And also there's other argument, I don't know if it's true now, there's been a theory around for many years that the core protein for the avian papillomaviruses is much longer. It may contain sequences that similar to what X does. So that's why we don't see X in viruses. So but that has not been totally resolved. I don't know if anybody still work on duck hepatitis virus anymore. So, but anyway, sorry, back there.
A very fascinating story about the permanent hepatitis B virus. I think you know very well in hepatitis C virus field, there's also a permanent strain called JFH, standing for Japanese permanent hepatitis C, and it has played a very important role in the HCV study field. I'm just curious, what do you think of the difference between two fulminant viruses, two different viruses, but they go fulminant, fulminant hepatitis, and other than viral factors, there are host factors involved.
Yeah, well, I mean, I'm absolutely in line with what you're thinking because I did a lot of my HCV work using this JFH1 strain, right? And was involved in some of the earlier characterization of these strains, and I mean, it's very rare fulminant hepatitis C. I mean, we actually have looked for it. We published a paper many years ago looking at all the fulminant hepatitis, so-called fulminant hepatitis, fit the definition to see whether any of them are related to C. This is, 20 years ago, we couldn't find. We must have 40, 50 cases. We couldn't find them. And so there are certainly sporadic report of fulminant hepatitis C, mostly from Asia, actually, interesting enough. I'm not aware of any fulminant hepatitis C in the US and Europe, and mostly in Asia. So I wonder whether there's something more to do, either the host or some environmental factors that that maybe it's not because of the virus itself cause fulminant hepatitis C. It's just because factor. And actually, I can tell you the JFH1 strain that many of us worked on is attenuated virus. I mean, we try to passage in chimpanzee, it causes very little infection, doesn't cause much disease. So it's totally opposite. And there's really very little phenotype with these strains that we use. Although it works well as a virus in cell culture because we can get high level infections, we can study the virus and use to study antiviral. And actually, that's been a very valuable tool for people working in the HCV field, right? This JFH1 strain. But it's probably not representative. I mean, actually, I can say it's not representative of all the HCV isolates. In fact, humans. It's a very unique strain. So that's what I think. So, yeah. That's all.
Jake, is HBV directly oncogenic once it's integrated, or is it just that it causes cirrhosis like other?
Well, probably both. I would think that probably the major driving force is activation of immune response, the injury, regeneration, set up a whole cycle, and then you have a mutation, introduced mutation during this high level of regeneration. But also there's some evidence suggests that HBV integration for both animal models and even human study, that integration probably does contribute a role, probably by altering the host gene expression profiles and non-specific. Certainly in the woodchuck model, it's very clear they integrate into the MYC locus, activate MYC protein. And there may be there's some evidence in the human HCC as well. It specifically integrates certain locus of either tumor suppressor gene or oncogene that could activate that. Also, don't forget about X protein, right? I mean, many people claim that's oncogenes. I'm not sure it really fit the classical definition of oncogene. Maybe Tom can comment on whether what is a classical definition of viral oncogene, but certainly for X, I don't think so. Sort of oncogene, but still, it has a pleiotropic role. That's what's considered a viral oncogene. It's not like the classical RAS, BRAS, or T antigens and all these other well-known viral oncogenes.
I have one more from online.
Okay.
Is HBe antigen essential for viral infection? Are E antigen negative patients infectious? If an E antigen negative virus infects another person, what happens? And will the virus revert back to E antigen?
Yeah, but in general, E antigen negative patients have much lower viral levels, right? Just the natural history. So probably they are less infectious, but you can certainly infect. There's no question about that. And I think what I show you with this outbreak of fulminant hepatitis B clearly shows the case that you have the chronic source, a patient whose source has a chronic HBV expresses contain the E antigen negative virus, who's able to passage to 5 other unrelated individuals and cause fulminant hepatitis B. So I think that's a clear example. There's a human transmission, and also this virus can be passaged in animal model, in the chimpanzee in the early days, and now certainly in our mouse model, right? We can show that clearly. They are equally as infectious as wild type virus. So yes, it is. And then the last question is that what is the consequence of if you're infected with the E antigen minus virus, pre-core mutant? I think that most of the clinical data suggests that you do get acute infection, probably more severe acute infection, but you don't get as much chronic infections as a wild type virus does. So that's kind of the general. Although it's not absolute, right? It's not that only virus infected with E antigen gets no chronic infection. It probably can, but it's just, just the difference is it's more of a quantitative difference, not a qualitative.
Thank you, everyone. We're gonna wrap up. Thank you so much again. Those of us online, thanks so much for joining. This will be recorded and posted in a couple of days on our website. And for those of us in person, there is a reception right out here in the lobby. So please eat, drink, you'll have a chance to talk with some incredible scientists and people in the room and have a chance to talk with Dr. Liang. But before we go out, just for everyone else, can I just get Jake and Randy and Tim for a photo real quick? Thanks, everyone.
Groundbreaking sauce.